Welcome, everyone, to this week's edition of the Extracellular Vesicle Club. I'm very pleased to introduce Agustin Enciso-Martinez, who is working with two groups in the Netherlands right now. And we have Rink Newland on the line as well. But, one of the big problems that we have in EV liquid biopsy is the needle in the haystack phenomenon. And that is that we've got all these EVs that are coming from platelets and red blood cells and other sources, and they may or may not contribute to our biomarker signature. So wouldn't it be nice if we had some way of just identifying or capturing those EVs that were going to tell us about the pathology that we're interested in? Well, today's EV Club, I believe, is going to show us one potential solution here. And as I was just speaking with Agustin about, he is actually one of the very few presenters on EV Club who has now presented twice, or will have now presented twice. So Agustin, thank you so much for being here and for sharing your work. And I'd like to turn the screen sharing over to you. As I'm doing that, as you're pulling up your slides, I just want to remind everybody that I'd like you to please put your comments and your questions in the chat box, and then I will call on you at the end. We'll allow you to unmute yourselves and show your video if you'd like, and have a conversation with Agustin and his co-authors. Agustin, please go ahead.
Thank you very much, Ken, for this opportunity. It is a pleasure for me to share our work, which was recently published in JEF. And it has been really a team effort between three institutes, the Leiden University Medical Center, Amsterdam UMC, and the University of Copenhagen. And this is about the identification of tumor-derived extracellular vesicles by targeting a unique and tumor-specific glycosylation. But let me begin with something that many of us are working to change. So a patient presented with vague symptoms, and after going back and forth with clinicians, they did a scan and took a biopsy, and unfortunately, he was diagnosed with pancreatic cancer. So the patient was put on a treatment, and after some time, it turned out that the treatment did not work. And in fact, the cancer had spread to other parts of the body. And this is a reality for many patients where clinicians miss the window opportunity to make an intervention earlier. Wouldn't it be nice, indeed, if we could detect the cancer even earlier before the symptoms appear? Or that we could follow it in time, the treatment response through the analysis of blood sample. And of course, many researchers are busy with this task. There is a whole field, the liquid biopsy field, where people try to identify particles that derive from the tumor directly in body fluids, such as the blood. So what can we find in the blood that comes from the tumor and that can tell us that a patient has cancer? Well, there are many particles, for example, circulating tumor cells, circulating tumor DNA. But we are very interested in the tumor-derived extracellular vesicles. These vesicles that are released by cancer cells in the tumor and that can enter the circulation. As they contain information about the tumor in situ, this gives us an opportunity to look at these pieces of information from the tumor directly in the blood. So we are very much interested in these tumor vesicles because they enrich information from viable tumor. Their presence in blood makes them relatively easy to access, and they could provide a blood-based window into the tumor composition and its dynamics. So they seem to hold potential as real-time biomarkers. But as Ken mentioned, for years we have been really struggling to detect and especially distinguish these tumor vesicles from many other particles in body fluids, such as blood. And one of the reasons is the lack of tumor-specific epitopes that enables us to reliably detect and isolate these tumor-derived extracellular vesicles. Besides that, vesicles are small, especially compared to cells, which translate into a low number of epitopes that we can use to target for detection or isolation. And if we put some numbers on this, we can see that an example just for a cancer cell, this is a prostate cancer cell with a diameter of 16 micrometers. This LNCAP cell will have around 600,000 molecules, one single cell. Now, if we assume the same surface density of EPCAM molecules, a vesicle of around 100 nanometers would only have 20 EPCAM molecules. So you can imagine that it is quite challenging if we are trying to detect it, for example, with fluorescent detection methods. So we label the vesicles. Because there are very few epitopes, many of these vesicles will appear dim or be under the detection limit of many standard detection assays. So the lack of tumor-specific epitopes or tumor-specific markers is a long-standing challenge in the cancer field. And this has led researchers to draw inspiration from other fields. And one such example is the remarkable parallels between cancer and embryonic development. So these two tissues, for example, tumors or the placenta, relied on rapid proliferation as well as extracellular matrix remodeling to invade tissues, whether it's the tumor invading adjacent stroma or whether it's the blastocyte invading the uterine lining. Another characteristic shared by these two types of tissue is immune evasion, which is central to the survival of both the placenta and the cancers. So researchers have shown that tumors can reactivate oncofetal programs so that they can actually re-express oncofetal markers. And one that stands out, or at least for us, is a post-translational modification called Oncofetal Chondroitin Sulphate, which has been shown to be shared in placenta and in tumors. And we were fortunate to begin this work together with our collaborators and co-authors, Alice Alanti and Mette Agerberg, who have previously identified this OFCS glycosylation on different types of tumors. So the glycosylation, OFCS, is attached to different proteins or proteoglycans. And it has been shown, as you can see in this image, that it is displayed in the tumor tissues, as you can see here in red, but not in the normal or normal adjacent tissue, except for the placenta, which is the only healthy tissue that displays this glycosylation. Now, this post-translational modification has been shown to modify or to be attached to more than 30 types of proteoglycans, and it can be selectively targeted by the malaria-derived protein called VAR2. And this has been previously shown by our co-authors, where you can see different tumor tissues from different cancer types in these images. And when they stain for the glycosylation using the VAR2 protein, which you see here in red, you see a very clear staining for the tumor or stroma, but not for the normal adjacent tissue. So we hypothesized that tumor-derived vesicles also display this OFCS glycosylation on their surface, and that we could target using probes that selectively bind to this OFCS. Now, we think that this has an advantage for the vesicles in the sense that the glycosylation is modifying different types of proteoglycans, so increases the amount of epitopes that we can target for detection, for example, unlike approaches that target a single protein. To answer the question whether this OFCS glycosylation is displayed on vesicles, which for us was key to identify these tumor-derived vesicles, we use some probes, that is the VAR2 protein, as well as antibody fragments that selectively bind to the OFCS. And we started just by first testing this on vesicles derived from cancer cell lines, and then we moved to plasma of pancreatic cancer patients. So first, what we did is just to validate the VAR2 labeling of cancer cells. And this is an example for the A549 lung cancer cells, where we try to really show the binding of the VAR2 protein to the OFCS present on these cancer cells. So we labeled them with a fluorescent VAR2 protein, the cells, and what we saw is indeed a clear staining of these cells with the VAR2, as you can see here in green. Now, to confirm the specificity of the binding between the VAR2 and the OFCS glycosylation, we included a negative control, and that is chondroitinase enzyme. So what happens here is that we treated the cells with this chondroitinase control, or ChAP-C. This enzyme digests these glycosylations, which means that the VAR2 would not bind anymore to the surface of these cells. And that is indeed what we see in this image. We also added another negative control, and that is just adding the fluorophore without the VAR2 protein to assess any possible non-specific binding. So we should not see a green signal or a fluorescent signal, as you see it in the first image, and that's what indeed I can see in this negative control. Then we moved to the vesicles, and we started by extracting vesicles from different cancer cell lines. We collected the conditioning medium, we removed cells and large debris. We concentrated the conditioning medium with tangential flow filtration, and then we captured the vesicles on a chip. And we captured based on a phosphatidylserine or by targeting tetraspanins on the vesicles. So how this works is basically we functionalize the chips with antibodies that, for example, target the commonly expressed tetraspanins on vesicles like CD9, CD63, CD81. And then we label with the fluorescent VAR2 protein. And of course, we did washes in between. And then we analyze this using fluorescent microscopy. And in this case, we use super resolution fluorescent microscopy, or DSTORM. For the first approach, where we attach vesicles based on this phosphatidylserine binding, we could see a clear labeling of VAR2, as you can see here in green, where each dot can show a single vesicles. With this technique, you can see in the middle plot the staining with the VAR2 or the green signal. The scale bar you see here is 100 nanometers. So we can really see the staining, and we also included the CD81 staining, which makes it easier also for us to look at the vesicles in our negative controls. So we identified three subpopulations of vesicles, one of them having only VAR2 staining, another one having both VAR2 and CD81, and a third one only having the CD81. But actually, the majority of the vesicles that we found there were only VAR2 positive. Now, we needed to, of course, confirm the specificity of the binding. So again, similar to what we did with the cells, we included this enzyme ChAP-C that digests the glycosylation. So we should see a decrease in the VAR2 signal, which is what we see here in the images, as well as here in the quantification. We also included the reagents control, and this is basically a procedural control where we have all our reagents, the VAR2 protein, the CD81, but no vesicles. And of course, the buffer where the vesicles were in. And we should also see a decrease of the fluorescent signal, which you also see here in the image and in the quantification. And this is, of course, important for the vesicles because antibody aggregates, for example, in fluorescent detection methods, they can give signals that just look like vesicles. We further validated this method, but now by capturing vesicles using antibodies against tetraspanins. So we can see again that after labeling with the VAR2, we see a clear VAR2 signal in this sample, and we also confirm the specificity of the binding between the VAR2 and the OFCS. In our negative control here is ChAP-C. You see the decrease of the VAR2 signal. We further confirm this specificity using now a mutated VAR2 protein, which shows a decreased binding to the OFCS. So again, we saw a decreased VAR2 signal, and that you can also see it here in the bottom in the quantification. And similar to what we did previously, we also added our reagents control or procedural control, where we also see a significant decrease of the VAR2 signal. Here you also we show some examples of single vesicles, some of them having both markers, some only having the VAR2 label. We also look at the CD81 signal, and we see that for our negative controls, ChAP-C and VAR2 mutant, we don't see a difference compared to our sample as expected, as the CD81 labeling should not be affected by, for example, the enzyme or the VAR2 staining. And in our reagents control, we saw a decrease of the CD81 signal, confirming that we have minimal antibody aggregates. Now we wanted to move to a to a non-biased way to actually label these vesicles with the VAR2, because so far we captured them based on the tetraspanins or the phosphatidylserine, but we wanted to label them in a non-biased way, and also to confirm this with an orthogonal method. So again, we isolated vesicles from different cancer cell lines. We use tangential flow filtration to concentrate them. And then we did the labeling in suspension. So we use the fluorescent VAR2 protein to label. We also include the CD81. And then we did size exclusion chromatography to remove unbound dye and also some soluble proteins there. We have also assessed the quality of our samples using an automated cryo-EM pipeline that allows us to take hundreds to thousands of images to really assess that we have vesicles, but also to see what else do we have in our samples, so to avoid having any particles that can be confounders in our analysis. So after we isolated the vesicle with the size exclusion chromatography, we then did the analysis with every scan super resolution microscopy, as well as with single vesicle flow cytometry. When we look at the microscopy result, we could validate the previous result where we showed the binding of the VAR2 to the vesicles. So we see a clear staining of the VAR2, which is nearly abolished in our negative controls, the SHAP-C, confirming the specificity of the binding between VAR2 and OFCS. And we also see a decrease of the signal in our detergent controls. Now, we added this detergent control to indirectly show that indeed we were labeling vesicles. So how it works is basically we added detergent, we treated the vesicles with detergent. These vesicles were labeled. In this case, we use MP40, and what happens is that then the vesicles lyse, and this results in the fluorophore being diluted in the sample. So for some detection methods, this actually means that the fluorescence signal will decrease because the vesicles are not concentrated anymore, the epitope, the fluorescent fluorophore actually. So we should see a decrease in the fluorescent signal, and that's also what you can see here in the microscopy as well as in the quantification for both of our negative controls. We further validated these findings using flow cytometry, where you can also see the clear staining of the VAR2 for the vesicles and the decrease of the VAR2 signal in our SHAP-C and MP40 controls. We again added the reagents control, where we could assess also minimal protein or antibody aggregation, as you can see here in these images. So after showing that this glycosylation was present, at least for these vesicles derived from these cancer cells, we wanted to assess whether the glycosylation of FCS is retained after epithelial to mesenchymal transition. And that is because in cancer, many cells, cancer cells of an epithelial phenotype, they undergo what is called the EMT. So they become more aggressive, migratory, so they acquire a mesenchymal phenotype. And during this process, some markers are lost. For example, EpCAM is lost during this process. So we wanted to assess whether the vesicles that are derived from these both phenotypes would still retain the glycosylation of FCS. So for that, we use lung cancer cells, A549, which have an epithelial phenotype. We induce the EMT using the cytokine TGF-beta, and we confirm the induction of the EMT with microscopy, where you can see the shift in the morphology of the cells from the epithelia to the mesenchymal phenotype, having more stretched morphology, and also with the staining of the actin fibers. And we also confirmed with this EMT with a Western blot, where we could see some of the upregulation and downregulation of some markers which are characteristics of EMT. We then isolated the vesicles from these two phenotypes. We stained them with VAR2, and we could see that indeed vesicles obtained from both phenotypes were stained with VAR2. And you can see it here in the quantification. So suggesting that indeed this OFCS is retained after EMT for these tumor-derived vesicles. And that we can also see a decrease of the signal in our enzyme control, this SHAP-C. So this supports the idea that we can actually target also the vesicles that come from different cancers or more of mesenchymal phenotype, and that could potentially increase the pool of vesicles that we can target in circulation. So far, I presented some results of these vesicles derived from the lung cancer cell lines, but we wanted to see whether this OFCS glycosylation is also present in vesicles derived from different types of cancer cells. For that, we isolated vesicles from cancer cells that come from melanoma, colorectal cancer, ovarian cancer, and glioblastoma. We stained them with the VAR2, which you can see here in green, and we saw a very clear and bright staining of these vesicles with the VAR2. And we confirmed the specificity of the binding of VAR2 to FCS, again with our ChAPC control, where we see that the signal is nearly abolished. And we also saw a decrease of the VAR2 signal in our MP40 controls, indicating that we were indeed labeling vesicles. You can also see this here in the quantification on the right. Now, these cells are mainly having more of a metastatic phenotype. So we try another cancer cell, more of an epithelial MCF7, breast cancer. And to our surprise, we could not detect VAR2 on these vesicles derived from these cancer cells. That's something that we are still trying to find out the reason of this. We then wanted to also test the VAR2 binding on some negative controls. So that is cells, basically epithelial cells, so non-malignant cells, and basically on their vesicles. So we collected their vesicles of these three types. So you can see here MCF10A, HMEC, and HACAT. And we observe a lower VAR2 signal compared to these other vesicles derived from these other cancer cells. As you can see it here, and also in the quantification, it's important to know that here the y-axis has the same scale for all these plots. However, we still could see some labeling of some particles, some background label of the VAR2, as you can see here, for example, for the MCF10A and the HMEC. We don't know the exact reason why we still see some of this labeling in these non-malignant vesicles. However, we have to take into account that these cells were actually immortalized, and they have been passaged multiple times. So this could also confer some cancer phenotype to these cells and vesicles, which could include, for example, the reexpression of some oncofetal markers like OFCS. So what we did next was to move towards more the blood and the plasma experiments. So what we did first is to mimic the plasma from cancer patients by basically spiking or mixing vesicles derived from cancer cell lines into healthy plasma. And once we mix this, we then label with the VAR2 to see if we could retrieve the signal of the VAR2. And what you can see here on the top left is indeed we were able to retrieve the VAR2 signal on these vesicles when we spike them in the plasma. And we confirmed the specificity of the binding with our negative controls, ChAPC. And also the VAR2 signal was also decreased with the MP40. And you can also see it here in the quantification. We then try different spiking ratios. We could also see the labeling, and still we could retrieve the signal of these vesicles with the VAR2, and we compare it with the labeling of VAR2 of only plasma that you can see here in the image as well as here. And we saw that in this case of when we label only healthy plasma, we saw a significant decrease of the VAR2 signal. And this was also validated further with flow cytometry. We then move to the plasma of pancreatic cancer patients. And these were the reason why we wanted to test this in pancreatic cancer. Well, there were many reasons. One of them is that clinicians there urgently need approaches that can help them get more information about the status of the patient. And there are many suboptimal biomarkers in pancreatic cancer. And another reason is that our university medical centers, the pancreatic cancer is a focus area. So what we did is we collected blood from pancreatic cancer patients, specifically pancreatic ductal adenocarcinoma, and also from healthy donors. We then isolated the plasma, and then we labeled with the VAR2. Also, we included CD81. We did SEC to collect the vesicle extraction and to remove unbound dye and also other soluble proteins. And then we also detected with the Eriscan microscopy as well as with flow cytometry. Now, what we first also wanted to check is just a pancreatic cancer cell line under vesicles, just to see we would expect some labeling there. And that's what we did first. We have here the PANC pancreatic cancer vesicles. As we could also see the VAR2 labeling to a lesser extent compared to other cancer cells. We saw again the specificity of the binding, where you see the decrease of the VAR2 signal in our negative controls for both the Eriscan and the flow cytometry. And then we move to a patient. And here I show an example first of one patient. So this is a PDAC patient where we saw a very striking difference between the plasma from the patient and the healthy plasma that both were labeled with the VAR2 fluorescent protein. And we saw a very clear VAR2 staining, also validated with both methods, the microscopy and the flow cytometry for the PDAC plasma. And a significant decrease for the healthy as well as for our negative controls, confirming the specificity of the binding between the VAR2 and the OFCS. So we expanded this to 4 more patients. These were advanced stage PDAC. And we could observe very similar differences between the PDAC plasma and the healthy plasma. We could observe that for 4 out of the 5 patients, it was a significant difference where we could see between the patient and the healthy controls. We saw a significant decrease of the VAR2 signal in our healthy controls as well as in our negative controls that I show here, in this case, the MP40 controls. So this first proof of concept study shows some promise in the detection of tumor-derived extracellular vesicles in the blood or in the plasma of pancreatic cancer patients. However, as you can also notice here in these healthy controls, we still see some background labeling on some VAR2 signal in some of these healthy controls. And if we are looking at pancreatic cancer patient blood in advanced stage with a high tumor load, then it's very clear that the difference will be clear, let's say, for clinicians between the plasma from patients and the healthy plasma, as you can see it here. But if we want to go for early stage cancer, the difference might be more subtle. And that means that we would like to have in our healthy controls basically a black image here, so no VAR2 signal, which so far we still see some signal there. So we try to assess whether we could decrease this background VAR2 signal by using, instead of the VAR2, a newly developed antibody fragment. This is called C9. It's a single chain variable fragment that targets also the OFCS glycosylation. And it has also been shown by our co-authors that the C9, which you can see here in red, also selectively labels tumor tissues, different types of tumor tissues, but not healthy or normal adjacent tissue. And we wanted to see whether this also antibody fragments would have a lower background. And with some pilot testing, and indeed seems that this is having a potential lower background than the VAR2 in the healthy plasma, but also using these antibody fragments again confirms the specificity of our staining that is really targeting the OFCS glycosylation. And I will just show you some very few examples of what we have done so far using these antibody fragments, C9. We here see vesicles derived from ovarian cancer, and we again saw very clear labeling of these vesicles, similar to the VAR2 in our previous experiment. In the CHAPSI control, we see a clear decrease of the C9, and also in the MP40, as you can see it also here in the quantification. These antibody fragments seem to be also promising for the labeling and detection of these tumor-derived vesicles. And I can show you also an interesting example here. These are vesicles derived from colorectal cancer cells. And it was interesting because we also could stain for EpCAM, as these cells and vesicles also express EpCAM here. And you can see here the merge image where you can see the C9 and EpCAM. But if we split the channels here and we look at the C9 and the EpCAM channel, we really see an increase labeling using the C9 compared to EpCAM. And I think this also shows an advantage of targeting this glycosylation OFCS in the sense that we increase the number of epitopes. So perhaps vesicles that are very small still have many OFCS epitopes that we could still detect, which will be more difficult or more challenging with targeting a single protein like EpCAM. And here at the bottom row, I also show the specificity, basically with the ChapC decreasing significantly the signal of the C9, but not affecting the signal of the EpCAM or the CD81. With that, I would like to summarize this study. So we have shown that this glycosylation OFCS is displayed on tumor-derived vesicles from cancer cell line and also detected in a plasma of pancreatic cancer patients. We also showed that the VAR2 and the C9 antibody fragments can enable the sensitive and specific detection of these OFCS-modified tumor vesicles. And OFCS seems to also be retained on the tumor-derived vesicles after EMT, at least for this model that we tested. So this could support its use across an heterogeneous tumor states and also increasing the pool of tumor vesicles that we can target in the blood of cancer patients. Now, I think this approach also enables selective isolation of tumor-derived vesicles. And because we have shown that these OFCS seem to be broadly present in tumor-derived vesicles coming from different cancer cells, it could potentially be used as a pan-cancer liquid biopsy. Now, moving forward, it's important to, of course, do further clinical validation, increase the size of the cohorts of patients used, as well as the stage. It is interesting to also see that just recently an independent group have also used VAR2 to target these tumor-derived vesicles in the blood of also pancreatic cancer patients. And they could also see the detection of these tumor vesicles actually across different stages of pancreatic cancer, confirming our findings and also being consistent with what we also observed. Now, this approach, I think it can also create opportunities for the selective enrichment of tumor-derived vesicles for either downstream analysis or for functional studies. So now we have a target that is OFCS, that is tumor-specific. So it's a handle that we can use also for this selective enrichment or pull down of these tumor vesicles. We could potentially also expand to other tumor types and assess different clinical applications. One that is very interesting is early cancer detection. Now, there the tumor load, and we expect also indeed that this concentration of these tumor vesicles in early stages is very low. However, if we have this OFCS handle, we could potentially use this approach to also enrich these tumor vesicles. So we can actually probe large volumes of plasma and enrich these small vesicles, or these rare vesicles that is we using this VAR2 or C9 targeting the OFCS. And I hope with this talk, I can show you how also interesting it is to target glycosylations on the vesicles, specifically these tumor-specific glycosylations like OFCS, because these are modifying different proteoglycans. So that means that we really increase the number of epitopes that we can use to target for detection and isolation. So that has an advantage for the vesicle field, especially comparing to targeting a single protein. With that, I would like to thank very much all the team that made this work possible. It has been really a team effort. I would like to thank very much Mette, Ali, and Caroline from the University of Copenhagen, Rink and Edwin from Amsterdam UNC, all the students that did a great job and were instrumental for this work. And also special thanks to Peter ten Dijke with the support and the guidance throughout this project. course, to the funding agencies and you for your attention. And I'm very happy to answer questions. Thank you very much.
Fantastic. Well, thank you so much, Agustin, for all of that great information. I just want to remind everybody, please put your questions and comments in the chat box, and then I will call on you in order of the questions that are received. But I would like to start with two questions of my own, one of them technical and the other one more biological. You mentioned that the PS capture was giving you a lot of background. And I think you showed some nice controls that you have done for your super resolution microscopy, a lot of great images. What do you think is going on with that? Why is PS giving so much background? You even chose to move to the tetraspanin capture instead.
Well, actually, the PS capture, we don't know exactly what that will be. In the reagents control, well, with super resolution microscopy, we indeed can see the background. I mean, this is a technique that can resolve for also a small diluted, basically dye that may be still attached to this. We, of course, isolated, these are vesicles isolated from cancer cells that were depleted from FES, and we also assess the quality. So in general, we think we have a good quality of having only these vesicles in these samples. So we didn't proceed further with the PS, but we really thought, okay, let's just go for the tetraspanins and see how that works. But I would say that the background was perhaps in the reagents control not so different between the tetraspanins and the PS.
Okay, yeah, that's interesting, because I think that in my group we've also seen quite a bit of background depending on which of the captures we're actually using.
Right.
So my second question is that if we subscribe to the idea that tumor-derived UVs in the circulation are also contributing to pathology and maybe contributing to metastasis, does your finding also open up the opportunity of doing some sort of blood filtration where you would be capturing the UVs and removing them in the case, I think, especially of advanced cancer?
Yeah, that is actually a great idea to have some sort of capture device that you can just circulate the blood from the patient, some sort of dialysis with a capture mechanism using for example, the nanowires that are targeting the vesicles. I think that will be indeed a great idea. Of course, there are still circulating tumor cells, but actually it has been shown also that these circulating tumor cells also display this OFCS. So maybe you can capture them both at the same time.
Yeah, no, I mean, it's been tried with viruses, right? To take viruses out of circulation, and some folks, I think, are trying to do that with UVs, but of course you have to have something that's fairly specific. Yeah. So let's move to the questions now that are in the chat box. And again, if you haven't put your question in the chat box, I'm happy to call on you. Just put a little note in there. So we also had a little bit of a problem in the last EV Club where people could not unmute themselves. So let's try a test case here. Johannes Thaler, I see you have a question. Could you unmute yourself perhaps? Yes. It's working today.
Do you hear me?
Yes, we can.
Yes. Great, it works now. Yeah, congratulations, very nice data, very interesting. I'm a hematologist at the Medical University in Vienna, so I'm not treating persons with solid tumors, but I have a bit of insight, and I would like to comment on your clinical conclusion. As you said, it could be used for early cancer detection or monitoring of a treatment. And I think the early detection could be quite problematic, because what you would need is a marker that is specific for an entity and sensitive, because and this is not the case here. And what you would have here is some indication that somewhere is some kind of cancer, right? So then you would have to do a whole body screening, and so this would lead to a tremendous amount of clinical workup, and this would be very cost ineffective and putting a lot of stress on the patients.
Yeah.
Yeah. And what I think would be more realistic is using this for treatment monitoring. So seeing whether these vesicles really go down following treatment, and if you could see treatment response. So in the next trial, it should be a longitudinal trial comparing pre-treatment and post-treatment and follow-up. This could be useful. And then I think it would be needed to compare it to current tumor markers. For example, CA19-9 for pancreatic cancer is, I think, quite usable. And it would have been necessary that it can compete with the current tumor markers or show some advantage.
Yeah, no, it's a very valid point. So for your first comment on the early detection, yeah, I mean, this seems to be a pan cancer marker, right? So yeah, if we detect some tumor signal, then this may come from any tissue, right, where the tumor could be. However, of course, this application in the early stage, we believe that it could be applied to specific clinical needs. And that is one of the ones that we are very interested in is pancreatic cancer and the detection of cancer in high-risk groups, right? So there are, for example, patients with some mutations, like, for example, the P16 mutation. These are people with an increased risk of developing pancreatic cancer. And they are enrolled in surveillance programs. So basically, they go every year to the hospital, they take an MRI and ultrasound to really track small lesions. This is suboptimal because maybe half of these patients could be missed. But if we could actually help clinicians with such an approach, where we could detect some tumor signals in the blood of these high-risk groups, maybe even earlier than what MRI or ultrasound can do, that definitely will help. And we think that these applications for early stage should be really indeed focused on specific cohorts that have, defined really clinical needs. Yeah, and for the validation indeed with the CA19-9 markers, that is, for example, used in pancreatic cancer. So that is something that just recently was also compared with in this last publication that I show, also where they also detected these vesicles using the VAR2, and they also compared with this CA19-9, and they found ROC curves also above 95%, let's say, of the area under the curve. So this is indeed very preliminary work. I think for now we have just shown these vesicles can display this glycosylation, but of course it requires further clinical validation and increased size of the cohorts and really assess specific needs indeed.
Thanks.
No,
Thank you.
It's great to have that perspective, Johannes, because we have to think about the patient, we have to think about the healthcare system and what it can bear, with these very early detections. I guess what Agustin has shown is this pan-cancer marker, and perhaps the possibility would be as well that should that signal be observed, if there is a sensitive assay to look for cargo, for example, nucleic acids, maybe we could even identify at least a potential source and not have to do the full body workup right away, as you mentioned. But of course, that requires a lot more work in very sensitive omics kinds of assays. Good. All right, let's go now to a question from Mohammed Niamat Hossein. Please go ahead. And I don't see him jumping on here, so maybe I'll just ask this question, and it's related to the healthy plasma versus TDEVs. How can you assure that the effect is due to the tumor-derived vesicles and not something that you find in healthy plasma? I guess you had those controls.
Yeah, indeed, we really compare with the healthy control, so we see this clear difference between the in this case regarding to the question the spiking and the healthy. We also see the consistent with the ratio. So when we have spike more of these vesicles, we see an increase of the VAR2 signal compared to when we spike less. And then in the healthy, we see a lot less signal. But yeah, it is true that in the healthy plasma controls, when we stand with VAR2, as I mentioned, we seem to have some background VAR2 binding. And the fact that this can be, even though it's minimal, still we see some fluorescent signal, as you can see in the slide. And the fact that this may be abolished using the CHAPC could indicate that this could potentially be chondroitin sulfate mediated. So not specifically to the OFCS glycosylation, but to the glycosylation chondroitin sulfate. And that is glycosylation that is not tumor specific.
Thank you. Susan Goals. Susan, you have maybe a follow-up here to the proposal to use this as a treatment monitoring strategy.
Yeah, I was, combining both yours and Johannes's comments. I was thinking about either cargo or surface specific molecules that mediate prognosis like metastasis, and thinking about whether or not you could early, you have a treatment that you say, okay, you've got stage 1, stage 2 cancer, we're going to give you a treatment and it's going to prevent, moving on to stage 4 cancer, whatever, prevent the metastasis. Could you use the EVs as a potential additional biomarker for potential treatment response, that kind of thing? And that could be, but it also could be a set of microRNAs inside the EV. So is there a way to, in your cohorts, that you basically isolate the EVs, then longitudinally, especially with something like pancreatic cancer, longitudinally figure out who, has metastatic disease, who doesn't, et cetera, and then begin to separate out the cargo that can predict that?
No, that's definitely a very good idea, and indeed, so our first analysis has been focused on the detection or basically of the concentration of these tumor-derived vesicles, right? And that is a motivation because we have seen, and it has been published, that the concentration of tumor-derived vesicles in blood seems to correlate with overall survival. So meaning that the more of these tumor vesicles in the blood, the worse the prognosis for the patient, indeed. So already only the concentration could give an indication, for example, if maybe a treatment is working or not. But indeed, the cargo is another added biomarker that we could look at that can also help for this stratification. Now, in that case, how I envision, for example, is to we have now a way to selectively pull these vesicles. Of course, it comes with a lot of challenges of enriching things from the blood, but we could selectively pull them using, for example, magnetic beads that are coated with the VAR2 or the C9 nano antibodies, and then we can really do the downstream characterization and see what else do they have that could help us for the treatment monitoring. And yeah, in pancreatic cancer, especially for a cohort of, for example, locally advanced pancreatic cancer, where at least in the Netherlands, they undergo neoadjuvant treatment to see if they can downstage and then really remove the tumor. Well, what we have seen is that many of these, or the majority actually of these patients that undergo neoadjuvant treatment without any significant benefit. So clinicians really would like to have a tool that can tell them whether the treatment is working, yes or not, in the early stages, so that the patient is not just enduring the side effects of the chemo without any significant therapy. And that's, of course, a lot of work that needs to be done, and this is just the beginning, but we hope together as a field we can look at all of these clinical applications.
Thank you.
Thank you.
Thanks, Susan. Next question is from Melissa Cuadron Lopez.
Hi there. Yes.
Hello.
Talk. Thank you. I am from McGill University, and I am on the side of malaria parasites. So I find fascinating that the VAR2 has been now found to be useful for something else, that parasite binding to the placenta. So my question is related to a technical one. And so how do you treat the EVs with the chondroitinase to remove the CSA? Do you do that in solution, or you do that on the chip? So how the treatment goes?
Yeah, that is in solution, in suspension. We add the chondroitinase, indeed.
Okay. Okay, thanks. And then the other question is, presumably people that is pregnant is secreting a lot of EVs coming from the placenta that might also contain chondroitin sulfate A. So
Yeah.
is there a limitation of this detection for applying this to pregnant patients?
Definitely, yeah. This would not work as such in pregnant women because their blood will also contain vesicles that are having OFCS, indeed. Yeah.
Do you guys have explored the presence of this chondroitin sulfate A in the EVs from pregnant people?
No, we haven't.
Okay. Okay,
Yeah.
thank you.
Thank you. Yeah.
And here's another idea for how this could be used. Minglin Liu, please.
Great work, great talk. And since chondroitin sulfate proteoglycan also appears in the atherosclerotic vascular wall, well, when the vascular you see in the unstable angina pectoris happens, coronary artery may rupture. That release, I'm sure, a lot of EVs as well as chondroitin sulfate stuffs. And have you guys think to expand from not only tumor also to the cardiovascular disease? That would be a great application since it's a cardiovascular disease and also a major death for the human.
Yeah, thank you very much. Yeah, so that would also, of course, depend on whether this would also spread the specific glycosylation that we look at, which is the oncofetal chondroitin sulphate. And the thing is, like all tissues display this chondroitin sulphate. However, there is a unique one that basically a unique glycosylation, a unique type of chondroitin sulphate that has a different sulfation pattern that makes it specific to cancer and to placenta.
Okay.
So I would not know if this will also appear during this type of cardiovascular diseases. Because so far, as far as I know, it's only restricted to cancer and placenta. This is specific modification of the CS.
Okay, good. Thank you.
Thank you.
Thanks for this comment. So it looks like we have come to the end of the questions that are in the chat box. Unless somebody else has a last minute question, I see that Emily says here, thanks for your talk, Agustin. I hope we could use it for hematological malignancies one day. So we'll see how this develops in the future and how this proposed use can maybe help some patients. So Agustin, thank you so much for the talk. I'd like to thank your co-authors as well for this great work. And I'd like to also thank everybody who joined us today and for all the good questions. So I hope everyone has a great rest of the week, and we'll see you again on a future EV Club. Thanks so much.
Thank you very much. Thanks a lot. Thanks a lot, Ken.